Thank you all for coming today for our exciting talk with Dr. Lee Hood. My name is Stephanie Adams, for some of you who may be coming from another school today to listen to our distinguished lecture. I'm going to introduce James Yurkovich in just a moment, who's going to introduce Dr. Hood. But I wanted to just let you know we have reconceptualized our distinguished lecture. So when I first joined, we had a distinguished lecture every month. And really what it was one department would nominate, but the departments have their own seminars. And so that we're not competing with each other, we decided to go to 2 distinguished lectures a year. And we will typically take suggestions on the topics, but we want the topics to be applicable to the entire school and campus. So we're not just going to focus on somebody's very specific research in a subject matter unless it's transformative across a lot of disciplines. So we will bring thought-provoking speakers about pedagogy. We'll bring people such as Dr. Hood, who have done exciting things in entrepreneurship as a result of their research and scholarship, right? So we're going to be looking for a different cadence of speakers. So I wanted to let folks know, as we're establishing our distinguished lecture, that it will be a little bit different than it was 3 years ago. So with that said, I am not going to introduce Lee. I'm going to introduce James. Dr. James Yurkovich is teaching what we like to call a first-of-its-kind graduate course here at UTD. The title is Systems Analysis in Digital Health, which is cross-listed with Systems Engineering and Biomedical Engineering. James is also on the Industrial Advisory Council for the Department of Biomedical Engineering. And with a PhD in data science and systems biology from UC San Diego, James has worked with Dr. Lee Hood for several years, first at the Institute for Systems Biology in Seattle and now at Phenome Health. They have published several papers together, so I thought it would be really more appropriate for someone who could fully give you the context of the impact of Dr. Hood's work to properly introduce him. So James, would you please come down and do the honors for us today?
Morning, everybody. This is a great honor for me to introduce my mentor. So I met Lee back in 2018. I finished my PhD, and I started a faculty fellowship at the institute that Lee founded in Seattle, the Institute for Systems Biology, and Lee was my faculty mentor. Worked with him for a few years. We published some papers together, and during that time, it was a true privilege of mine to be able to work with one of the pioneering technologists, biotechnologists of the last several decades. Lee's lab over the last 20, 30 years has been responsible for developing a lot of different really crucial engineering technologies that have opened the way and paved the way for many different fields. His labs developed the automated DNA sequencer, the automated DNA synthesizer, the peptide synthesizer, and protein sequencer, all of which enabled a lot of the technologies that we use today and even some of the consumer products. Along the way, Lee's lab has spun out a lot of these technologies to be able to optimize them and bring them commercially to scale up the technologies. He's been involved in the founding of, I think it's over 17 different companies, including Applied Biosystems, Amgen, and Aerovale, and most recently Phenome Health, which is his latest startup that I have joined, and a lot of what he's going to talk about today is really in this era of phenomic science. So we have the genome and we have the phenome, and all of what Lee's going to talk about today is a vision for what the future of medicine looks like when we bring all of this data together and we have true data-driven medicine and science. And so with that, it is my great honor to introduce Lee, one of the most determined optimists I've ever met, and I think that's the one thing that he can pass down to me most. So everybody, thank you. Welcome to Lee.
Thank you. Thank you. Can you hear me okay? Good, good. Well, I'd like to really talk about my entire career and how it is really focused toward this data-driven approach to health. And in fact, I remember back when I started at Caltech as an assistant professor in 1970, thinking, as assistant professors do, where do I want to go in the future? And A real interest in human biology and human disease. I'd just gotten my MD at Johns Hopkins and so forth. But what caused me serious concern was the enormous complexity of humans and the fact there were no good tools for attacking biology of humans where it should be attacked. That is, to generate enormous amounts of data that hopefully could be used in deconvoluting all of this complexity. So what I'm going to talk about is the outcome of that thinking that led to this idea of we're going to, in the hopefully not too distant future, totally replace healthcare now focused on disease with a healthcare focused on wellness and prevention. And I'll divide this lecture into 3 parts. I'll start by giving you a historical context and a conceptual context of how I think about these kinds of issues. Then I'm going to tell you 6 stories from the data from a population of 5,000 people we looked at longitudinally over a 4-year period. And each of these stories illuminate another aspect of what health-driven care in the future is going to be. And then finally, I'll talk about where we're going to go in the future. So starting with the history, at Caltech, these considerations of biological complexity led me to a course of action that went through 7 paradigm changes, as I'll call them, 7 kind of fundamentally new ways of looking at biology or doing biology. These all dealt with complexity, but they framed beautifully what data-driven healthcare should be, so I'll mention those very briefly. And the first you've already heard from James. We brought engineering to biology over a 25-year period by actually developing 6 instruments that allowed us to read and write DNA and proteins in various ways. And these were exactly what we needed to develop to begin looking at big data in individual human beings. One of these instruments got me invited to the first meeting ever held on the Human Genome Project in the spring of 1985, where 12 of us went together and were asked the question, is this practical? What are its limitations? And the conclusions we came to were 2. 1, it is practical, though at that time very difficult because, for example, you needed to develop a whole series of new chemistries for doing the fluorescent dyes we need to do DNA sequencing. And number 2, we were split 6-6 on whether it was a good idea. Those opposed, because big science would take, so they argued, all the money away from small science. And of course, we engineered a way so that argument got eliminated entirely by persuading a congressman from New Mexico to make a pitch for $3 billion of new money to go into biology. I will say, in the beginning, DOE drove this program and did a beautiful job. NIH was opposed to it all the way up to the very end when it started in 1990 and so forth. And of course, it was completed in 2003, and for me, one of the most important aspects of it was, for the first time, we had access to human variability and the ability to correlate that with either wellness phenotypes or disease phenotypes, and we'll talk about that later. I made a pitch in the late 80s to biology at Caltech that, look, the next evolutionary stage of a biological department would be to make itself cross-disciplinary, to bring in scientists from all the different disciplines so they could invent the technologies and the strategies we'd need to go forward in both in biology and in human medicine and so forth. And this endeavor was shot down entirely by the biology department. And Bill Gates made it possible to go to the University of Washington, set up the first such department. And without going into it in any detail, over an 8-year period, it was marvelously successful. For example, Phil Green invented the 2 key software programs for the Genome Project, and Rudy Abersol and John Yates started the field of proteomics at that point in time. But what was clear to me by the end of that century was a big state bureaucracy really makes it difficult to do something strikingly new in biology, and that was systems biology. And I can tell you 10 different ways I was frustrated, but in the end, I resigned from the University of Washington to start the Institute for Systems Biology, which has for the last 22 years, 3 years, been very successful in pioneering systems approaches both to biology and disease, and as we'll see in just a moment, to wellness and so forth. And of course, the Institute for Systems Biology pioneered this thinking now, which is holistic and global, and it is really key to be longitudinal in nature because you need to reconstruct biology from the data that assays at relevant points in time the biology that you're trying to understand. And that's what systems biology brought in spades. But I'll also say that systems thinking toward healthcare led us to the conclusion that it should be predictive, preventive, personalized, and participatory, so-called P4 healthcare, and that there were 2 domains of this new healthcare. One was disease, which then, in the early 2000s, about 99% of healthcare was focused on, and today, maybe we can say 96% of healthcare is focused on. And the other, of course, was wellness. And it was in 2013 and 14 that Nathan Price, who had then moved to the Institute for Systems Biology as a data expert, and I formulated the idea that we could actually begin to look in a data-rich sense at a population of 108 of our friends by doing a complete genome analysis, followed by analytes in the blood, proteins, clinical chemistries, and metabolites, followed by the microbiome and a Fitbit for digital health. And we did this for a period of 4 years. And this generated the data that really all the stories will come from. And they validate the approach of phenomics that we're going to be talking about in just a few minutes. And of course, that led to Aerovale, where we as a company brought scientific wellness to consumers and acquired 5,000 customers over 4 years. And you'll hear all about the data there and everything. And the final step in my career and in thinking about this whole endeavor was when Rod Hochman, CEO of Providence, a very large healthcare system in 7 states, 51 hospitals, came to me and said he wanted me to become chief science officer at Providence and to bring scientific wellness and to bring genomics to Providence. And this seemed like an incredibly exciting opportunity. And for 5 years I was there. I learned about the healthcare system in a way I couldn't have from the outside. And I came to the conclusion that what we needed to do next in the trajectory I've laid out for you was to get a population 200 times as great as 5,000, 1 million individuals, which we would look at in what is going to be a second genome-like project over a period of 10 years to do genome-phenome analysis and to validate the snapshots that I'll talk to you about in just a few moments. So what I do want to emphasize is the whole outlook of analyzing this data is really driven by systems thinking. And the essential fundamental point of view was this idea that blood is a window into health and disease. And it is so because it bathes all organs. They secrete molecules into the blood. And if you can read the origin of those molecules and quantify them, you can draw conclusions about the health and disease state of these various organ systems. And we've done that very beautifully. And of course, the data that we got from proteins and metabolites and clinical chemistries could be converted into classic digital kinds of information. And then it was translated into and mapped into the biological networks that are the core underpinning of physiology and disease in a human being. And this ability then to move to understand the dynamics of the coordinated behavior of these data is what is giving us really big insights into fundamental aspects of health and disease. Now, I just want to emphasize that the genome was a $3 billion project which over a period of 12 years generated one genome sequence. And the Human Phenome Initiative that I'm instituting proposes to do 1 million individuals and do their genome and phenome analyses over that period of time. Of course, the phenome of a human is nothing more than the different state changes that you exhibit as you develop and age across your entire lifetime. So what phenomics does then is samples at appropriate periods of that lifetime your blood and your microbiome and your digital health and things like this. And it gives us insights from 3 areas. The phenome constitutes a contribution from your genome, from your behavior, and from your environment. And those can be assayed by these samples that we've talked about from blood and microbiome and digital health and things like that. And it's that information that we use to generate the Aravale approach to things. Now, what this led to was, of course, the ability to understand in enormous detail an articulation of precisely what the vision for the future was. And that is this idea that we could use a data-driven analysis to assess each individual's health trajectory and to optimize that health trajectory, filling in inappropriate kinds of ways. And of course, what was interesting, there we go. What was interesting about the health trajectory is it showed, number one, that there were 3 phases of that health trajectory. We were born in wellness. Almost all of us transition 1 or more times to disease. The transition is the 2nd phase. And then the 3rd phase is the disease and its progression, moving generally to termination and back to wellness and so forth. And in 2 mouse models, one for brain cancer and one for brain degeneration, prion disease, we demonstrated beautifully, because we could initiate the disease at a given time point, that the number of disease-perturbed networks associated with disease progression increased exponentially as disease progressed. And that means you want to be able to detect disease at the earliest stage and reverse it when it is very, very simple. So these 3 stages then we've used in the Aravale population, the data-driven approach, to, on the one hand, be able to optimize your wellness. I would say most ordinary well people have 20 or 30% of their wellness potential. And we can, with data, create actionable possibilities that will allow us to optimize this strikingly for each individual. And of course, the real question is, to what extent do you want to participate in this whole process and contribute to it? Where it is really important is when you get to be middle-aged and older, because then you can hope to expand significantly your health span as opposed to just your lifespan. You can move into your 90s or even hundreds, mentally alert and physically capable and the like. We were able to show in the Aravella population that we could detect these early disease transitions to chronic diseases up to 1 to 4 years prior to clinical diagnosis. So that means we have a tag that says, let's develop therapies for reversing these early stage diseases before they manifest clinically. And there are lots of ways to think about doing that. And finally, you could use the precision medicine techniques to be able to attack disease itself and take this very powerful data-driven approach. And we'll do that for diabetes a little bit later in this whole course and everything. So again, your health has these 3 states: wellness, transitions, and disease. And I would argue that they will lead us to this revolution of replacing wellness and prevention for the current disease focus we have today. And I think this will be the biggest paradigm change in healthcare that has happened in the last 2,000 plus years or so. Finally, I want to just say that we have really good preliminary evidence that argue this data-driven healthcare approach is going to work. And of course, one comes from the Airvale population of 5,000 over 4 years. A second comes from something I haven't talked about, but in the project we'll be pushing forward. We're going to look at brain health in the context of being able to make digital measurements that assess 25 different cognitive features, and where they've become diminished, give you the potential to replace lost function. And what's exciting about this is this lost function can be regained even in 80-year-olds, as a study of 1,000 individuals showed. And what was remarkable is it showed that the majority of 80-year-olds could go back to cognitive levels close to what they approached at their maximum in the early 30s, mid-30s, and things like that. So there's hope for all of us. Anyway, and then finally, we've used the data from Aravale, and many others have used it too, or used other kinds of data, to show the gut microbiome plays really a major role in health, and we'll talk a little bit about that and give you some examples of things that we've discovered. I'll tell you that almost 30 papers now have come out of the data from Airvale. And again, each of them tells a story that illuminates a different aspect of what health is going to look like in the future. And I'm going to go on and give you the 6 stories that I think illuminate some of the most important features. And the first of these is how we can actually detect these transitions very early, and that is going to lead to, hopefully, the prevention of chronic disease. And this all started with a single woman who we noticed in 2017 had been diagnosed with stage 4 pancreatic cancer, which is really a grim diagnosis, unfortunately. But we had the idea of asking, since we had about 6 blood draws prior to her clinical diagnosis, could we see the detection in the blood of proteins that mark the transition from normal to this disease? And when we did that analysis with about 500 different proteins, we were able in this woman to show one protein in particular really stood out as abnormally high compared to all of the other control individuals we had at that point in the study. And in fact, we found 4 more proteins that fell in this same category. 3 of those proteins mapped beautifully into disease-perturbed networks common to stage 4 pancreatic cancer. The other 2 were unknown proteins that we had nothing about. But what this suggested to us was an exciting idea is that we could detect chronic diseases. And when we looked back carefully at the entire Airvale population, we detected 167 transitions from wellness to many different chronic diseases. 35 of those were to cancer, and we took 10 of the cancer patients and did exactly the same kind of analysis we talked about here and showed in every single case we had proteins that marked the transition to various different types of cancers and so forth. And I think what's really exciting is in the million-person project over 10 years, we predict we'll get 200,000 transitions. And of course, these will give us more than enough data to be able, at the initial identification of the transition, to predict just what disease we're going to, and then to be able to set up and methodically develop therapies that can reverse these things early. Now, if we're going to be successful, that's a big if, it'll take a lot of work to do it, it will be the beginning of the end of many chronic diseases. And I'm very optimistic about that kind of possibility. So that is an example of prevention, okay? Story number 2 is scientific wellness and actionable possibilities. And of course, these came very nicely from the fact that we used the classic tools of, one, generating an enormous amount of data. In this case, it's the 108 pioneers that we did in 2014 for a 9-month period. And we were able then to take the data from each of these individuals and so forth and carry out traditional statistical analyses that showed which data pieces in the 6 different data types that we had there would be statistically correlated with other data types. And we could then take interesting correlations of different data types like this and go to the literature and actually discern actionable possibilities, that is, led to things people could do that would either improve their wellness or let them avoid disease. And with the Airvale population, we came up with the order of 200 or so of these actionable possibilities, all focused on wellness. With the larger Phenome Initiative million-person project, we think we'll come up with more than 10,000 new possibilities. And here, AI will be utterly essential for delivering to the doctor 2 things. 1, what you should do for this actionable possibility, and the evidence that supports it. And both of those things are key, and I think can uniquely be delivered in the actionable possibilities. But this is all to say that, we have an enormous number of actionable possibilities we can consider from the different data types, and that the 2 types of wellness we can define map beautifully one to another. So there's traditional wellness, and there are 100 companies out there that are looking at diet and exercise and stress and sleep and things like that. And of course, our data-driven approach, looking for actionable possibilities. From the genome will come thousands of new actionable possibilities, some that have been defined by the Medical College of Human Genetics, and others that will be defined by things called rare diseases and Mendelian recessive diseases, of which there are 7,000 each in human beings. And of course, the other set of actionable possibilities of interest to most of us here there are hundreds of variants that correlate with athletic injuries. And if you know you have that variant, you can do exercises to avoid ACL tears and things like that. So there are many possibilities at the genome level. At the phenome level, there are likewise many possibilities and so forth, and we'll talk about those. But where a lot of possibilities come is in the integration of different types of data. For example, my own experience was my vitamin D level was very low in the 100 Pioneer project. And the only thing that brought it up to normal level was excessively high levels of vitamin D, not 1,000 international units, but 15,000. And the reason for that is I had two variants in my genome, each of which blocked the uptake of vitamin D that could be overcome by excessively large amounts of vitamin D. And I have to maintain myself at a normal level with 5,000 units of vitamin D a day. But the really important point is that data-driven approach allows us to go back to conventional wellness and to specify uniquely for each individual what aspects of exercise, diet, sleep, and so forth they especially should look at. So we can bring personalized medicine to what is now classically formulated as population medicine. Everybody does the same kind of thing. Okay, so that's actionable possibilities. It's going to create wellness and so forth. And stubborn. So story 3 is about genetic risk and differential treatments for people that fall in high risk or low risk categories. And let me start out by saying, gosh, this is going to be a pain. Maybe it'll work, yeah. That the variants within the human being that average 1 in 1,000 base pairs or so, called the polymorphisms, of course, are the substrate for doing genetic correlations of phenotype with individual things. And I wonder if we can get another advancer, because... Oh, okay. I would say pointing at the computer underneath. The computer's underneath the cabinet. It doesn't tap, does it?
James, the computer's in the cabinet. Maybe and the dongle's down there, so point it down there.
Oh. Doesn't do anything. I think maybe the battery's down or something like that. Oh.
Yeah, he'll do the keyboard.
Oh, I can do the keyboard. Does the keyboard work?
Yep.
Oh, okay. And that's, okay, good. Well, anyway, with the polygenic scores that are being generated now for 150 disease phenotypes or wellness phenotypes, people are looking at hundreds of thousands of individuals to identify the variants that each contribute in very small ways to this. And one of the classic examples is height. And they've recently done a study where they've got 180,000 variants that collectively contribute to 60% of the height determination. And they're quite confident that most of that is going to be genetic in the end, and with another 180,000, maybe they'll get up close to 100%. But most of the diseases that we look at here, you're lucky if the polymorphisms explain 20%, but that's still sufficient for making the kind of calls that we've got here. So from the polygenic scores, we then can take individuals whose genome sequences are known, and we can assign you high risk or low risk or medium risk, whatever whatever else you are and everything. So when you look at these polygenic scores and you look at a population, and this is just looking at 4 different types of diseases, doesn't matter what they are. The conclusion that is very strongly true is the upper 5% of individuals in the genetic risk score should be treated essentially as if they have the disease, and you should do all the preventive things that you can think about for them because they're just at the point of transitioning, so you want to get them before they actually transition. But we can make one point even more strongly about this, and that is, this shows the population from Aravale from low risk to high risk in 5 pins. And it shows the fact, if you look at the individuals that aren't taking any kinds of medicine for anti-cholesterol, statins and things like that, the level of cholesterol goes up linearly with the increased genetic risk. And the really striking observation we made with the Aravale population is for people that have high genetic risk and high cholesterol, only statins will bring down the cholesterol. If you look at people with low risk and high cholesterol, diet and exercise bring it down beautifully. And I'm going to argue that for virtually every chronic disease, we're going to treat high and low risk people differently. So now we, as I said, can do roughly 150 of these or so. And I think this is going to be a major opportunity for actionable possibilities in the future. Story 4 is all about aging and health span. And here the story is really interesting. We were able to show in the Aravale population, as you aged, and we had a population that ranged from 21 to 93, as you aged, you lost successively the ability to control the expression of all 3 of the categories of blood analytes we looked at, proteins and metabolites and clinical chemistries. And we could actually use that to develop an algorithm, and I'm not going to go into any detail about how we did it, but that algorithm allowed us to determine for each individual your biologic as opposed to your chronologic age. The lower your biological age is relative to your chronological age, the better you're aging. And what we showed in the Aravale program that was really striking is for every year over the 4-year period an individual stayed in the program, they lost a year to a year and a half of biological age. A striking decrease in this delta change that meant they were aging much more successfully. And one of my closest friends lost 10 years of biological age in this whole process. So it raises questions about how we can slow the biological aging process. And I think we're going to be able to slow it in really striking ways. The other point that I would make is that individuals who had various diseases in the Aravale population, in every single case, had biological ages higher than their chronologic ages. And with diabetes, it was 6 years higher than their biological age. And in fact, if you looked at people in the upper 5% of Fitbit activity, you could show their biological ages were 3 years below the average of their compatriots that didn't have disease. So it was really quite striking to say, this is a metric for wellness. And what I think was even more important was to realize not only could it give you your global biological age for your entire body, but for major organs like the kidney and the liver and the immune system and lipid health, metabolic health, we could get biological age scores. And because we were using metabolites to determine the biological age, we could make inferences about how to decrease the biological age of organs and or the global biological age if it were too high. So this metric gives us the ability to actually suppress even further biological ages. And that turns out to be a really important point because aging is the highest risk factor for all chronic diseases. So if you can slow the aging process and at the same time make people scientifically healthy, then you're going to have powerful tools for avoiding chronic diseases. And that, coupled together with detecting them early and reversing them, puts us in a wonderful position to avoid chronic diseases in the future. And that turns out to be important because of our $4 trillion cost in healthcare, 86.6% of it is devoted to chronic diseases. And suppose we could eliminate half of them in 10 years, which is not an unreasonable estimate of what we might do. You're talking about, close to $2 trillion. And of course, the interesting question is where would that money go? But that's another debate. So story 5 is about metabolic BMI. And as you know, metabolic BMI is a proxy for the conventional BMI, which is determined by your height and weight. And it's very good at predicting chronic diseases and things like that. But it is very, very inaccurate for about 30% of the people. And we've actually had a paper just recently last week in Nature Medicine that showed that we could do a metabolic age which was enormously more flexible in responding to changing times. We could actually look into the 30% and actually classify them. One example is very muscular athletes almost always are obese by the conventional BMI, and we got it exactly right in these different cases. So I think the metabolic BMI will replace that, and it is a wonderful metric along with biological age for being able to follow the health of individuals, quite obviously, and to do it in an easy, very accurate fashion and so forth. And the final story is about the gut microbiome and health. And as you know, this has gotten a lot of publicity in the last 2 to 3 years as being a fundamental component of health that has to do with diet and all sorts of things. And the essence of what one wants to strive for in the microbiome is diversity. Diversity equals good health in many different ways. And almost all the diseases of the gut microbiome lead to enormous restrictions and diversity. And we did, again, from the Aerovale patient, 3 really interesting things. One, we could show that there were 11 metabolites in the blood that very accurately predicted the gut microbiome. And the reason that is really important is it's an independent check from gut microbiome diversity apart from doing sequencing of the gut microbiome itself. And it gives us the ability to go back and look at bloods where samples were not collected from the feces to be able to do the microbiome and reconstitute microbiome diversity. And you can do beautiful correlations with things in that way. And the actual third thing that we did that I found was really fascinating is we looked at individuals that were 60 to 85 to 88 years of age. We looked at about 9,000 of them, so it was a very big study. And what we were able to demonstrate beautifully is if you were a healthy individual, when we followed your gut microbiome, 2 really important things happened. One, you lost the earlier constitution of your gut microbiome species. And 2, each healthy individual uniquely developed a gut microbiome spectrum different from everybody else's. And the argument was that was in part responding to the needs you had as a 70 or 80 or 90-year-old and so forth. Now, in contrast, if you looked at people that were unhealthy, neither of those things happened. You maintained the early gut microbiome profile, and you didn't individualize in any way, shape, or form. And what was particularly interesting when we looked at 80-year-olds across a 4-year period, we were able to demonstrate the unhealthy people died 4 times as frequently as the healthy people did. So that kind of confirmed the supposition and the change that we were looking at healthy and unhealthy. And of course, the really intriguing idea is, can we facilitate healthy aging by facilitating the individualization of your gut microbiome as you age? And there are ways to think about that in very interesting ways. So anyway, those are 6 different pictures of the things we can do, and they show you a data-driven approach to health that is all about the science of wellness and the science of prevention. And we've shown you multiple examples of how we can bring those to play and so forth. So what is exactly next in this Phenome initiative and so forth? And we've taken 2 different tacks. In the short term, what we'd like to do is use a very data-rich approach to the 4 major chronic diseases. We're starting first with diabetes. We hope to have that funded within months or so, and start on a study that I'm going to describe in just a few moments. And then second, of course, we're already actively, and have been for the last 1.5 years, lobbying Congress to have, if you will, a second genome project where we look at 1 million people over a 10-year period with genome-phenome analysis. And we prove in spades all of the things that I just talked to you about with the preceding 6 stories. And I could give you another 12 or 14 stories if we had the time to go through them. So there's an enormous wealth of opportunities there. So Phenome Health is a nonprofit that we started about 2 years ago. James is a key member of the innovation officer in Phenome Health and everything. And to tell you where we're about now is to tell you that we have 6 different partners that uniquely make the Million Person Project possible. One, Guardian Research Network, is an organization that has access to, soon it will have access to 50 million different patients. They've learned with natural language processing how to extract electronic health records that include both structured and unstructured data, doctor's notes, and so forth. And this gives us the possibility of, and they've already considered the patients for experimental research projects, exactly like the Million Person Project we've designed. And this is where we're going to draw our diabetes population. And I'll tell you more about that in just a moment. But the really important thing is this organization interfaces with about 150 hospitals in the South and the Southeast, and they lie across minority regions, including African Americans and Latinos, and diversity is really a key part of the project we'll be talking about here. Phenome Health is the one that's going to partner with us on the brain health that I actually talked about. Technicity is actually a very small company headed up by B.J. Yurkovich that has actually helped us, together with James, build a multi-omic computational platform accessible to all 3 different types of data clouds, and with infinite capacity for extensibility to bring in whatever data we're going to need to bring in the future. Google we're collaborating with, hopefully on hyperscale AI, and I'm going to talk about that right at the end of the lecture in a few different slides and so forth. ISB has faculty members that are expert in knowledge graphs and expert in digital twins, and we'll be using both of those in the context of the diabetes project I'm going to talk about. And I'll say we'll be using both of those to create a digital twin for wellness too, but that will come a little bit later and so forth. And then finally, we've collaborated with Scientific American to make a 75-page article that discusses the essence of the new science of scientific wellness. And it's been a terrific introductory and persuading opportunity to bring in other partners and things like this. And we're probably going to be doing another one on health span and aging, topics I've just been talking about here. And we've used philanthropy to set the Phenome Health up initially, and we'll be hopefully getting $25 million from any one of a couple of different sources, hopefully in the next few months to get started on the diabetes project. I'll really stress how important racial diversity in the data are. This shows you the response of 3 different populations to ApoE4 homozygosity. That is the most striking predisposition for Alzheimer's that there is. And you'll see that the Japanese are about 3 times as likely to get Alzheimer's from the risk point of view as Caucasians are, and the Caucasians are about twice as likely as Africans are, and Hispanics almost have no reaction to homozygous. And it says really interesting things about Alzheimer's mechanisms, which we haven't got time to talk about. But Alzheimer's is really a disease of metabolism, and it's really a second diabetes in many ways. That's the underlying bottom line. Here is the diversity of phenomic measurements we'll be making in this project. They include many additional blood measurements. They include, for the first time, the brain measurements that are so critical, the microbiome measurements and things like that. And they also include the digital health records, both for body and for brain, and electronic health records, patients' outcomes, and social determinants of health, as well as complete genome analysis. So these individuals will have 10 times as much data gathered on them, roughly, as the Aravale population we had before. And I'll tell you that factor of 10 is going to make an enormous difference in using hyperscale AI, and I'll tell you why I say that in a couple of minutes. So what is next in the Human Phenome Initiative? It's this short-term approach to a diabetes project we've talked about here. And I'll just say that the 4 major chronic diseases we want to attack are shown here. Alzheimer's, we want to be able to attack heart disease. We'll probably look at heart failure. And we're going to look next at a cancer, acute myelogenous leukemia. For a lot of technical reasons, it's an ideal cancer to study with a deep data approach. And now we're going to be looking at diabetes. And what we're going to do with diabetes is look at it in 3 separate stages of data-rich analyses and so forth. And the key points that have to come out of all of these studies are, for a given chronic disease, how many different subtypes do you have? For example, diabetes, it's 4 or 5, most people agree today. And can we define those subtypes well? They aren't well defined at all. And the second point is, within a given subtype, you go through a series of state changes as the disease progresses. can we find biomarkers for those state changes? And an example is when there are a subset of individuals with diagnosed diabetes that go on to get chronic kidney disease, and that is society's most costly disease right there, with transplantation and dialysis as necessary outcomes. So can we find that transition and block it from ever happening? That's kind of question that we're really interested in. So the threefold approach, we've already analyzed 10 million electronic health records from a relatively normal population, which has 11% diabetics, approximately 30% pre-diabetics, and the rest normals. And here we're looking at markers that will let us, A, subtype the major types of diabetes, and B, to follow within a given subtype these different transition points. And once we have those, and we have some nice ones now, we'll use those to select populations, for example, that are early in the diabetes process, intermediate in the diabetes process, and more terminal in the diabetes process. And if we divide those patients into 3 or 4 different classes, and we plan to do about 250 patients for each, we'll be able to run 4 clinical trials in parallel for a period of 4 years and get the entire natural history of diabetes, which normally takes 15 to 20 years to follow. And once we've learned from data-dense prospective studies following these patients, then we'll go to clinical trials that are interventional in nature, where we'll study biomarker candidates and drugs that will make a difference and so forth. So this threefold approach is what we'll use for each of these different 4 chronic diseases. And nothing like this has ever been done before. And I'll make the bold statement that in 4 years, we'll learn more than we have in the last 50 years about diabetes. So, and of course, one of the key things about diabetes is that for the diabetic medicines, you only see a fraction of the individuals that are able to respond. And the reason for that is these 4 different subtypes, and each of them is going to require different drugs. If we can classify them, then we can begin to look in each subtype for the drugs that can deal with that effectively. And what I'm going to say is we'll see this for all 4 chronic diseases that we look at and the rest of the chronic diseases out there. But there are going to have to be entirely new methods for very large-scale discovery of new drugs. And none of the pharma out there have the faintest idea how to do this. And we have a company called P4 Bios that I don't have time to talk about, but it's going to be using peptide drugs, and it can generate easily up to 10 to the 12 different peptides and select from those in ways that haven't been used before. So we think we can have a pharma company that in time will match the needs that we have here. And the other point I'd make, and I know James disagrees with this slide to a certain extent, it was published in Nature 4 or 5 years ago now, and it made the argument, if you look at the 10 top diseases, 10% of the people or less respond to drugs. And those responses range from 1 in 4 individuals to 1 in 25 individuals. And of course, that means 90% of the drugs are wasted. In the first few years of the genome-phenome study, we'll be able to get biomarkers that will match beautifully all drugs with those who can respond. And in principle, you could save 90% of 600 billion. But let's say these is not accurate and it's 30% or 40%. You can choose whatever you want. I would argue you're going to have an enormous savings in drugs this way. And of course, pharma is going to fight this all the way because it'll hit at the bottom line of the same drug for everybody. And of course, they're starting to change this point of view, but with enormous reluctance. So what really is next in the Human Phenome Initiative, of course, this million-person project, and the point that I'd make here is it's really going to answer these 6 major problems of the quality of healthcare we have now. We're at the bottom of the top 20 nations with regard to developed and so forth. And the whole question of aging, the whole question of chronic diseases, the whole question of increasing costs, racial diversity, and a very different style for carrying out wellness and so forth. Those are all absolutely key facts. So do we have to quit now? I have one more major point I'd like to make, and we'll talk about this. The data that's going to be required to look at the problem of how do we convert the enormous phenomic data into an understanding of the disease and where to go is illustrated here. And you can see, well, this is, first of all, this is 2 knowledge graphs that we'll be using to acquire data in the hyperscale AI system. And it shows that Google really has lots of edges and major points, whereas Spike is a knowledge graph from UCSF that's been beautifully, very beautifully annotated and everything. And of course, this shows you the amount of data that is required from one blood draw from one person. And you can see the staggering amount of data that are required. And what I'm proposing to do in the future is to replace this thing I gave you before, where you take the data of individuals and a population, and you get the statistics that will lead to actionable possibilities. And instead, what we'll do is use these 2 knowledge graphs and bring them into a digital twin for diabetes, where from the top down we have physiology and differential equations that express mechanisms, and the bottom up biochemistry and omics and clinical chemistries. And thus this will be an enormously educated digital twin that will put in, for a portion of the million-person project, the individual data and everything, and we'll use that to educate essentially a ChatGPT equivalent of hopefully, if we agree with Google, to create an educated hyperscale system that will have the ability then to feed in individual data. And some would argue not only can you go to incredible instructions on what's missing and what's increased at high levels and so forth, but you may have the data to go directly to causal inference and avoid the statistical block that occurs in the more conventional kinds of studies. And that's a question that's up for incredible debate. So with that, I think I'll close and be glad to answer any kind of questions. So.
All right, thank you very much, Lee. I think we're a little bit over time, so maybe 1 or 2 quick questions. Yeah, go ahead.
Thank you so much, Dr. Hood, for this talk. Yes, I have one question and one very small question. My question is, you mentioned when you were talking about aging, you mentioned a concept that I can directly relate to something like age is relative. That's what I understood from what you said. And I would like to know, how do you measure that relativity of age? Because you say that chronological age is very different from biological age. So how do you measure that difference? And my smaller question is a personal question. You played football in high school. You spend your entire career fighting the status quo, pretty much. And I would like to know, what are the 3 characters that we can learn from you personally? And if you can recommend a book and a movie.
Well, I was going to talk about the book I just published that I would really encourage you, if you're interested in this thing. It covers everything I talked about and goes way beyond many things I talked about. But if you ask me I think, so my biological age is about 15 years less than my chronologic age, okay? And I think the major features for me that have contributed to that have been exercise. So I've been a fanatic about exercise most of my life, and I still am. And I think intermittent fasting is a terrific way not to gain weight. I mean, when you go into society today, it's really hard not to be critical of these people that are 20 pounds or 30 pounds overweight and don't know it. And it's going to have an enormous toll on you as you age. And your health span is going to be shorter than your lifespan is, frankly. I would say the other thing that I think has had a big impact on me more recently is balancing a lot of the vitamins and nutrients and supplements in the blood and things like that. And I think that has added further to this age differential. But the other thing you can do is use this biologic metric to show that indeed you can drive people's age down beautifully. And if you want to really understand how we derive the biological age is enormously complicated. And there's a good paper that lays it all out beautifully, and you should read that, because I could give you analogies, but in the end, empirically, we looked at a whole series of different potential algorithms and chose one that operationally worked really well to give us the kind of data that we've got. And it gives slopes that are absolutely parallel to your regular chronologic aging slope and everything. And you want that. You want to see those be in sync, but the one setback below it as far as you possibly can. So.
All right, we got time for one more quick question. Dr. Blaris?
Thank you for your presentation. This is amazing and honor to have you here. Rumors are that you do 100 push-ups per day.
I actually do 150 a day.
150, okay.
Yeah.
I see, okay. So I guess my question is, for faculty that are just starting their careers or young faculty, what specific problems or topics in this arguably big area would you suggest that they focus on?
I think the most exciting topic in this project, unequivocally, is data analysis. So don't get caught in generating the data. That's workman's tasks, and we can get people to do that. But the data analysis still, and it's not just turning the crank, it requires that the data analysts understand biology deeply and put that knowledge into the assessment of the data analysis and so forth. But look, the million-person project is going to be 200-fold as much data as we had in the Airville project. So, I mean, there's enough there for, I'd argue there's enough there for 100 really good data analysts, or maybe more, to analyze. I mean, the Airville data, I mean, every time you sit down and think about it, you come up with a new kind of conclusion of where you should go and what you should explore and what you should do. So I think, so that's one thing. I think the data end of things. I think the other things is the small biology that lets us end up checking and going from statistical correlations to causal inference is really going to, this is going to present small biology with an infinity of possibilities and opportunities and things like that. So if you're on the lab side of things, you'll have an awful lot to do too. And I think what you want to do is try and get the lab side situated at least in general proximity with the data side, so you can have access to all the latest data and figure out what types of hypotheses you'd be interested in doing. So I think both lab and data analysis are going to offer really unique opportunities. And the genome has really illustrated that in a lot of beautiful ways. It's taken a lot longer to develop than many of us thought it would, but it's starting to explode with information, with rare genes and things like this now. So, but the phenome will be a whole nother scale of things, because when you integrate different types of data, you approximate the reality of human complexity much more than if you look at a single dimension of data. And that's, I think, what's exciting about the genome. And I think there are just so many different ways you can do the analyses that are just waiting for people that are creative and energetic and innovative, that it's going to be, I think, marvelous opportunity.
All right.
Sounds
please,
like that's the end.
Yeah, thank you again to our speaker. And I'm going to hand it back to Dean Adams now.
Thanks, James. Dr. Hood, oh my gosh. You did exactly what a distinguished lecturer is supposed to do. You awakened our brains. I am so energetic and jazzed right now. I've got thoughts, I've got jokes, I've got political commentary.
Well, if you've got jokes, I could use some.
I mean, just the things that you said. I mean, I was trying to drop my notes down. They're all over this piece of paper. Right? So let me just try to make some points here. So first of all, I want to thank you for reinforcing things that I've been thinking for so many years and have been trying to communicate. One is that engineering isn't everything. Even though you're not an engineer, you're the son of an engineer. So I'm the daughter of a biologist and you're a biologist. So somewhere between us.
I argue operationally I'm an engineer.
Okay, well, then we'll give you that.
I'm a really ignorant engineer.
But it did, when I thought about some of the topics that you raised, it's some of the things that we're doing here in the Johnson School. You talked about AI, you talked about big data, you talked about brain health. I see my good colleague, our fellow dean from Behavior and Brain Science here today. You talked about systems thinking. I think every engineer needs systems thinking from the day they come on campus as an engineer, and Steve and I talk about that all the time. Oh my gosh, you talked about, as I said, I didn't have time to organize my notes. You talked about the fact that diversity is key. And in a state where we keep talking about we shouldn't talk about diversity, the numbers and statistics that you put up echo why we have to have diversity and why we have to have it in science and why we have to have it in representation in everything that we do. So thank you for that. Let's see, there was something else. The joke I had for you. So you talked about being able to avoid tears in the ACL. Where were you 40 years ago when I tore my ACL the first time, right? And then maybe 20 years later when I tore it the second time, right?
Yeah.
Now I have a new knee. I can't tear it anymore, but I could use it for the other one that I have that's not torn. How about that?
You can.
Okay, good.
You can.
All right. Then you brought up, is B.J. Yurkovich another relative of yours? So, like, is there like a Yurkovich-Hood relationship going?
Well, I think
Three Yurkoviches in a hood. There's got to be a joke there.
I think the Yurkoviches are going to take over the world.
Okay. Well, I'm going to get on that team because I think the world of Steve. So, Steve, thank you for bringing Dr. Hood to us. Thank you, Provost Musselman, for coming today. I know she's a very, very, very busy schedule. We've got a full day today with investiture this afternoon. So, for her to come, your topic was just amazing. And we can't thank you enough for, as I said, we were rethinking how we would do distinguished lectures, and you've set the bar really high. So now we've got to go back and think about who the next person's going to be, because I wouldn't want to follow you. Thanks to everyone who came today, students and faculty, staff that are here. One of the things I just wanted to say that I heard him say, that I took from his talk for everybody in the room is keep dreaming, keep pushing, and get out of your comfort zone. I mean, we can't keep staying in silos. The work that we have to do is really transdisciplinary, and I think you've echoed that. So thank you for coming. We have a small token. It is nowhere near what you gave us today. I am really full
No,
in
thank
knowledge.
you.
It was a
Thank you
pleasure
so
to visit
much.
with you this morning. I wish we had more time.
Right.
Join me in thanking Dr. Hood for coming today. I think I hit all my notes.
Wow.
Okay, so Steve, we are dismissed? Yes. We are dismissed. Thanks, everyone.